The result came back at 2:47 in the morning, and Dr. Elena Vasquez stared at the screen for a long time before she said anything at all. Not because she didn't understand what she was seeing, but because she understood it perfectly, and that was the problem. In 15 years of ancient DNA research, she had never seen a genomic profile like this. Nobody had. And in that quiet lab in Santiago, surrounded by humming equipment and the soft blue glow of monitors, the history of humanity had just, quietly, cracked open. For decades, ancient DNA research has been one of science's most powerful tools for rewriting human prehistory. And until recently, researchers believed they had a solid working map of where we all came from. The field exploded in the early 2000s, when scientists first successfully extracted and sequenced genetic material from ancient remains. What followed was extraordinary. Migration routes confirmed, population mixtures revealed. Lost peoples, entire civilizations that left no written record, suddenly given a genetic voice. By 2020, researchers at institutions like the Max Planck Institute for Evolutionary Anthropology in Leipzig had built perhaps the most comprehensive ancient human genetic database ever assembled. Thousands of samples, hundreds of populations. A map of prehistoric humanity so detailed it could trace the movement of a single ancestral group across continents over thousands of years. The assumption, reasonable, evidence-based, widely accepted, was that we had accounted for the major branches of the human family tree. Not all of them, certainly, but the broad architecture, the main lineages. Scientists walked into labs with confidence. They knew what ancient DNA looked like. They knew what anomalies looked like. They knew the difference between contamination and a genuine genetic surprise, or they thought they did. But here is the thing about science at its best. Sometimes the data refuses to cooperate with your confidence. The mummy had been sleeping in the high desert of northern Chile for approximately 4,800 years before anyone disturbed it. The Atacama, the driest non-polar desert on Earth, a place where rain is measured not in inches but in decades, is a remarkable preservationist. Bodies buried in its soil do not decompose in the way bodies buried elsewhere do. The extreme aridity essentially freeze-dries organic tissue, hair, skin, internal organs, even, critically, DNA. The specimen recovered from a site near the town of San Pedro de Atacama in 2019 was, by all initial assessments, a remarkable but not unprecedented find. A female individual. Estimated age at death, between 35 and 45 years. Burial goods suggested she held some form of social significance within her community. Carefully woven textiles, ceramic vessels, the arrangement of her body indicating deliberate ritual preparation. Anthropologists from the Universidad de Chile, who conducted the initial excavation, noted that the burial was consistent with pre-Columbian Andean funerary practices of the period. Everything about her. The isotope analysis of her teeth suggesting she had lived in the region her entire life. The wear patterns on her joints indicating years of agricultural labor, the specific style of the burial shroud. Everything pointed to a woman who was, in the context of her time and place, entirely expected. A member of an ancient Andean population. A thread in a tapestry researchers already thought they understood. Genetic sampling was authorized as part of a broader archaeological study. Nobody anticipated what was coming. The samples were sent to 3 separate laboratories, one in Santiago, one at the University of Copenhagen, and one affiliated with Harvard Medical School's ancient DNA program. Standard procedure, redundancy to guard against contamination. Results were expected within 8 to 12 weeks. They came back in 6. And everything about this burial had said ordinary. The DNA said something else entirely. When the full genomic sequence came back, Dr. Vasquez's team did what any competent scientist would do. They assumed they had made a mistake. This is important to understand, because it speaks to the rigor with which this anomaly was eventually confirmed. The first response to an impossible result in science is never excitement. It is skepticism. It is the quiet, methodical process of elimination. Could the sample have been contaminated with modern DNA during excavation? Ruled out. The extraction was performed under sterile field conditions with full personnel genomic exclusion protocols. Could degradation have corrupted the sequence? Ruled out. The preservation state of this individual was exceptional, and the sequence quality metrics were among the highest ever recorded for remains of this age. Could there have been a laboratory processing error? Ruled out, because Copenhagen and Harvard returned the same result independently. without knowledge of each other's findings. What all three labs found, what they could not explain away, was this. Approximately 7.3% of this woman's genome did not match any known human population, ancient or modern. Not a slight statistical deviation, not a marginal outlier within an established haplogroup. A substantial, coherent, reproducible genomic signature that corresponded to nothing. No known archaic human admixture. Not Neanderthal, not Denisovan, the mysterious ancient humans identified through a finger bone in a Siberian cave whose genetic fingerprint now appears in populations across Asia and the Pacific. Not the recently identified ghost lineage detected in West African populations. A deeply diverged hominin that interbred with modern humans perhaps 50,000 years ago and left traces in contemporary genomes. This was different. The genetic distance calculations placed this unknown component further from any identified human population than Neanderthals are from us. Let that land for a moment. Further than Neanderthals. Here is where the story becomes genuinely difficult to explain. And believe me, 3 independent teams of geneticists have tried. The first explanation researchers reached for was a migration model. Perhaps this woman's ancestors represented a founding population that arrived in South America via an entirely unknown route, not the Bering Land Bridge crossing that accounts for the ancestors of virtually all indigenous American populations, but something else. A coastal route, a trans-Pacific crossing, some deeply ancient movement of people that left no other genetic trace on the continent. Professor James Adler, a population geneticist at the University of Copenhagen who was part of the team that analyzed the second sample set, stated in a paper submitted to Nature Genetics in late 2023, a paper that underwent an unusually prolonged peer review process, that, quote, the simplest migration model cannot account for the divergence we are observing. The component in question would require a separation time from the main human lineage, exceeding what our current models of modern human dispersal from Africa can accommodate. He paused in his written remarks before adding, and this is the part that stays with you. We are not prepared to state what this implies. Dr. Miriam Osai Bonsu, a specialist in archaic human genomics at the Smithsonian's Human Origins Program, who has spent 19 years studying genetic evidence of ancient interbreeding events, reviewed the published data and offered a more cautious interpretation. She suggested the anomalous component might represent admixture from a deeply diverged population of modern humans. Not a separate species, but a lineage that had been isolated for an extraordinarily long period, long enough to accumulate this level of genetic divergence. Possible, in theory. But when the divergence timelines were modeled under this hypothesis, they produced numbers that pushed uncomfortably against the edges of what we know about when and how modern Homo sapiens left Africa and spread across the globe. The math worked. Barely. And only if you accepted starting assumptions that mainstream paleoanthropology has not yet endorsed. The interbreeding hypothesis was similarly strained. For admixture to explain a 7.3% genomic contribution, the interbreeding event would have had to be substantial, sustained, and involve a population that has left, inexplicably, zero other genetic trace in any of the thousands of ancient and modern samples in the global database. Zero. Not a whisper, not a fragment, just this one woman, in the Atacama, 4,800 years ago. Now here's something that doesn't get discussed in press releases. The Nature Genetics paper submitted by Professor Adler's team in late 2023 did not appear in print until nearly 14 months later. An unusually long delay, even accounting for the rigor of peer review for findings of this nature. During that interval, according to researchers familiar with the process, the paper underwent no fewer than 4 separate rounds of revision requests. Not for methodological errors, which were found to be sound, but for what reviewers described as issues with the framing of the implications. 2 members of the extended research consortium quietly withdrew their names from the author list before publication. When asked about it in an interview with a science journalist in early 2024, one of those researchers, who asked not to be identified, said only that they had concerns about how the findings would be received. A second anomalous specimen with a similar, though less pronounced, genetic signature was identified from a site in coastal Peru in 2022. That finding has not been published. It remains, as of this recording, classified under a heritage protection designation that restricts public data access. Why? That question has not been answered. Perhaps it is bureaucratic, perhaps it is procedural, or perhaps someone looked at what was in that second genome and decided the world wasn't ready for the conversation. What this genome may represent and the word may carries enormous weight here, is evidence of a human population so isolated, so divergent, that our current models of prehistoric migration simply cannot account for its existence. Not a different species, perhaps, but something on the threshold. A branch of the human family that separated early, very early, and lived in isolation long enough to become, genetically speaking, almost unrecognizable, and then vanished. Leaving behind, in one woman's bones, preserved by the driest desert on Earth, a message written in the language of DNA that we are only now beginning to have the tools to read. But not yet, it seems, the courage to fully interpret. Dr. Vasquez, in a rare public comment made at a symposium in Buenos Aires in March 2024, said something that has stayed with researchers who were in the room. She said, "Every time we think we have drawn the complete map of who we are and where we came from, the past finds a way to remind us that the map was always smaller than the territory." So I'll leave you with 3 questions that the scientists themselves haven't answered. Who was this woman, really? Not her name, not her role in her community, but what lineage did she carry, and where did it begin? Why does a second specimen exist, with similar anomalies locked behind a heritage restriction that prevents scientific scrutiny? And, if this genomic signature represents a population, however small, however ancient, where are the others? What do you think is in that data? Option A, a statistical anomaly that further research will eventually explain within conventional models. Option B, evidence of a human lineage so ancient and so isolated that finding it forces us to redraw everything. Leave your answer in the comments. I read every single one. And if this investigation has unsettled something in you, good. That feeling means you're paying attention. Because the truth is always stranger and older and more complicated than anyone is prepared to admit.